welcome you all to the technical session 4, where we dive into some cutting edge discussion on how technology can pave the way for a more sustainable future. To begin, I would like to extend a warm welcome to our esteemed session chair, Professor Dr. Pankaj Kumar Mishra sir, whose guidance and vision have been instrumental in advancing research in technological innovation and environmental sustainability. Thank you, sir, for being us today with us. Also, I would like to welcome our invited speaker, Dr. Hemant Nuthlapati sir, whose work in application of AI-assisted Raman spectroscopy has greatly contributed to the field of sustainability. It's an honor to have you, sir, with us. We are looking forward to your insights and valuable contribution. Now, a special welcome also goes to our participants, both those who are joining us here in the person and those who are attending online from across the country. Your enthusiasm and dedication of exploring sustainable solution are what make this conference a success. Today's technical session promises insightful presentation and discussions on various aspects of technological innovations that drive environmental sustainability. We will hear from researchers and experts who have dedicated their efforts to the developing sustainable technologies and methodologies to combat pressing environmental challenges. So thank you all of your enthusiasm. We continue this technical session with our focus, and our chairman is Professor Dr. Pankaj Kumar Mishra sir, and who's distinguished academic 22 year of experience in teaching, research and administrative experience is very well known, and he currently serves as a professor of applied physics, the University of PhD coordinator at Amity University, Madhya Pradesh. He is a gold medalist in his MSc. He completed his PhD in physics from the Durgavati University, Jabalpur, and he is also a DSc at Barkatullah University, Bhopal. Dr. Mishra's research span various domains and within the physics as well as in the electronics, with the expertise in electric and thin film technology in microelectronics. His impressive publication record includes around 75 research papers in SCI and Scopus Index journals and conference proceedings. He has also authored a book on engineering physics, filed 15 patents, and completed a research project funded by Madhya Pradesh Council of Science and Technology. He is an active contributor to the research community, serving a guest editor of Material Today proceedings. Pankaj sir, am I audible right now?
Yes, madam, you are audible.
Pankaj sir, am I audible right now?
Yes, ma'am, you are audible.
Okay, sir. I thought there is a pin drop silence, so thought it's not audible.
No, you are
So
speaking
today,
extremely well, so all are listening.
thank you, sir. He's an active contributor to the research community, serving as a guest editor of Material Today proceedings. He's a chief editor of the International Journal of Life Sciences and Pharma Research and a reviewer for various reputed journals. Many of his research students have successfully completed their PhD under his guidance and contributed towards the advancement in microelectronics. Today, he will be presenting topic, but before that we are going to start with the our plenary section with the Dr. Hemant Nuthlapati sir. Sir, are you ready for your presentation?
Yes.
Okay, sir. Okay, sir. So, as we continue our plenary session, it is a great honor to introduce our speaker, Dr. Hemant Nuthlapati, sir, who will be delivering a plenary lecture on application of AI assisted Raman microscopy from single cell to diagnostics. Now, I want to give a brief introduction about our invited speaker. He is an esteemed assistant professor at Raman Center for Medical and Biological Applications at Shimane University, Japan. Originally from southern India, Dr. Nuthlapati pursued his master's degree in chemistry from Banaras Hindu University, Varanasi, before receiving the Taiwan scholarship from the Ministry of Education to complete his PhD at National Chiao Tung University, Taiwan. His research, their focus on applying Raman spectroscopy to single cells is very good. And following a postdoctoral fellowship in the ultimate spectroscopy and imaging laboratory at NCTU, is he joined Shimane University in 2014, and doctor having groundbreaking research focuses on using Raman spectroscopy for the early diagnosis of diseases such as cancer, and harnessing AI to enhance diagnostic accuracy. Beyond his scientific contribution, he is actively involved in fostering Indo-Japanese collaboration in higher education and research, strengthening ties between premier institutions in both the countries. Dr. Hemant sir, we are honored to have you with us today, and we are looking forward to your valuable insights. The platform is yours now.
Thank you very much for your kind introduction. In fact, so without wasting any time, let me just share my screen and start the presentation. Yeah.
Yes, sir.
Okay. So let me know if we can see my screen.
Yes, sir. Screen is visible now and you are audible. Perfect.
And thank you. Thank you very much.
Okay.
You get a pointer. Okay. So, okay. So today, so my name is Hemant Nutalapati. I'm from India, of course. I've been, as kindly introduced already, I've been working in Shimane University. It's a national university in Japan, which I'll briefly introduce. But today, our topic for discussion is how we can, apply, develop AI-assisted Raman spectroscopy, especially to study single cells to diagnostic applications. So primarily, biomedical applications is our main target in my laboratory, but also I think I'll explain to it later. So, well, this is my email ID if any of you need to contact, and this is our emblem. And of course, right now everybody knows that almost every institution across the world is somehow trying to contribute to the SDGs, which is for the common good, global good. So we in our university also promote many research teams and many research centers focused on these sustainable development goals. And my presentation today, especially this national conference, is on sustainability. And we are trying to, in my lab, trying to focus on number 3, which is good health and well-being for all. So many of you may not know where is Shimane University. So let me give you a very short introduction. So this is Japan. Probably you know where is Japan, the north kind of east part of the world. And here is Tokyo. You know Tokyo very well, probably. And also Osaka is here. And Hiroshima is here. So you can see Shimane is in the west part of Japan. And Shimane has a very long coastline. This is Sea of Japan, Japan Sea on the west. So Matsue City, I'm just showing up this part, enlarged here. You can see Matsue. Matsue is the capital city of Shimane State, right? So I'm right now here. And we have 2 large campuses. One is in Matsue, another is in Izumo, somewhere here. But what is so special about Shimane? Well, population in Japan is graying and aging. So this state is one of the least populated, second least populated state in Japan. In fact, the reason I'm telling you this is also to invite most of you who are interested in Japan to come and study or work in Japan in the future. So the whole state, if you can probably may not believe, only has about 640,000, means 6.5 lakhs people living in the whole state. And it's a very large area, but the population density is very, very small, with about 100 people per square kilometer, right? And also, in terms of area, it's about 20 out of 40. There are total 47 states in Japan, and it's a very large area with very low population. However, this region traditionally is very, very important culturally speaking, and also for the origin of Japanese language and culture. So I just list several major points. In this state, we have 2 large UNESCO World Heritage Sites, and we have national treasures. We have one of the best gardens in Japan, and some of the best, not the best, I should not say the best, but most important shrines or temples are also here. So this is also considered, this region is considered to be the origin of Japan in Japanese culture, in Japanese folklore, right? So I hope you can someday visit and enjoy peaceful Japan. Well, let me very briefly also introduce what I do in my laboratory. What are my goals, right? So primary thing is about spectral and imaging big data, both acquisition of big data and also analysis of big data. You can just think about it in just those two words. And how do we do that? We try to develop new methods, new imaging methods, new spectroscopic methods in order to acquire a lot of data from various samples. Okay, that is one thing we try to automate. And again, we try to combine our expertise, that is in Raman spectroscopy and Raman microscopy. We build our own microscopes. We would like to combine and with other techniques to make multimodal technologies, which can help get alternative information in addition to the molecular information that we can get in Raman spectroscopy. For example, digital holography or other ENOs or something like that. And we are always hungry for various new applications, novel applications. Of course, we start with biomedical applications for disease diagnostics, pathology, pharmacology, food and agriculture applications, and also environmental applications, which I'm not discussing today due to lack of time, but mainly I'll focus on these one or two results. And inadvertently, almost everywhere, wherever there is data, we now know already it's been very well established. We need to do some sort of analysis, otherwise it's almost impossible. So we have been traditionally doing lots of multivariate statistical analysis, which is also a part of, a little small part of AI. But of course, statistics, yes, we do both univariate and multivariate statistics, but that is not sufficient, especially if you want to take any, decisions. So, and also automated decisions. So we, of course, make use of AI or machine learning, depending on the question we have. So today I'll be introducing a few examples, 4 or 5 examples maybe, on how we can study Raman's, single cells, or how we can apply these multivariate statistical models, how we can get very new molecular information. And I think, I should have introduced one more slide. I'm very sorry. I did not introduce Raman spectroscopy as such here. Oops. Okay. So what is Raman spectroscopy? Raman spectroscopy is nothing but a simple analytical technique for which is also a molecular spectroscopic technique, which gives very, very specific molecular information from any sample that we measure. And Raman spectrum traditionally is also called as a molecular fingerprint. So that way, most of the other techniques cannot give you that high specificity, but Raman spectroscopy can give that specificity. So if you combine Raman spectroscopy with a microscope, then it becomes Raman microscopy, where you are able to actually measure or probe very, very small, tiny samples. For example, like single cells, bacterial cells, it could be animal cells or tissues, or any sample that is, not, of course, at the microscopic scale, right? So what we can do, I'll quickly give you an example where you will automatically understand what kind of data we can get, right? Let's say first example is label-free molecular imaging of fungal cell or spore wall. So most of you must have heard about yeast, or simply talk about some sort of fungus, right? Yeast is a fungus, and they have very specific, architecture. And so the cell walls of yeast are very complicated, and they are made up of so many different kinds of polysaccharides, which are structurally very similar, yet so different. So let's say if you zoom up in the schematic, it shows that the cell walls are made up of several kinds of glucans, proteins, or some mannans, and then also there's other possibilities. Like glucan is not one kind of molecule. We have alpha glucans, we have beta glucans, we have mannans, we have these are all made up of either glucose monomers or mannose monomers and so on. Then what happens is that if you see, these are literally very, very similar, but not identical, because only the bond here is different, right? So because of the change in this bond, it becomes a totally different molecule, and the properties are also very different. And most of the traditional techniques that we use to study single cells cannot really differentiate such tiny, differences. However, Raman spectroscopy being molecular spectroscopy, which is very sensitive to study molecular structures, even we challenge that it is possible to study or differentiate between such closely resembling, molecules. So, anyways, no one has actually, uh, you know, studied in the living cell condition with all these cells or imaging of these various components. So we set out to do Raman spectroscopic, develop Raman spectroscopic method to study these polysaccharides. How we can do that? So in case of reproduction of, or the life cycle of fission yeast, there are, either sexual reproduction or asexual reproduction. Asexual reproduction is very, very common, especially very well known, also pretty well understood. Where it's a fission yeast, so it goes through medial fission in the center. So the DNA is dividing and then moving to two different edges, and then this is called as mitosis, which is very well studied. However, under unfavorable conditions, it can also undergo sexual reproduction that is called as meiosis, where two different individual cells will come together and mate in order to make what is called as four spores. So these are spore formations, and we have, not only the cell wall like that, which is fused cell wall, but also some new membranes are formed, which are shown in black here, which are called as spore walls, right? So these are four spores within one mother cell. So why are these important? Because spores are especially relevant because we have spores in actual air. I mean, everywhere in the environment, right? Only when the conditions are favorable, it can just spring back to life. And then, if it's infectious, then it can create trouble. If it's non-infectious, that's a different matter. But anyways, they are existing almost everywhere. Not only this fission yeast, of course, it's not like fission yeast is everywhere. For example, bacterial spores are there. So many, other spores are also normally there in the environment, right? So how do we actually study? As I mentioned, all the studies have to break these cells, try to separate it, and then try to only look at, glucans or how much is there, or, traditionally you can apply, mass spectrometric methods, chromatography plus mass spectrometry, where you can separate them by literally breaking them apart. But you cannot actually see them where they are. That means you totally lose spatial information. So that's why we want to develop some sort of an imaging technique, right? Let's say if you, it's a laser spectroscopy where we will simply use a single laser and of any color for that matter. And then we are trying to look at the scattered light, right? So for example, in our setup, we have a laser which is as tiny as, let's say, 1 micron, and that can give us a spatial resolution of about, say, 500 nanometers. Then if you just look at the scale bar here, it's 5 microns, right? So if you just measure, probably the pointer that I'm using is the actual size of the laser in the microscope that we have, right? So then what happens is, if you just measure the point A, you get Raman spectrum like this, shown in blue, which we can say that there are several bands that indicate the presence of lipid in this particular point, right? But if I do, measure, the measure point B, then we get totally different Raman spectrum, right? The same way, if you get a spore, you make a spore and then measure, it looks like completely different Raman spectrum, right? So by looking at all these, by interpreting these, what we can see now, we want to identify various polysaccharides, right? So this is the polysaccharide spectrum that we got from this black one, from spore. And we can compare with so many standards, right? For example, there are several beta glucans, there are alpha glucans. Alpha glucans also may have different kinds of, branching, especially with 1,3 branches or 1,6 branches and so on. So you can measure all of these and try to identify some sort of marker bands that are indicative of alpha or beta structures. And what we found is, well, so already you see 893 is a beta marker, 944 or 550, these are alpha markers. and 424 is a non-specific marker for polysaccharides. And so now we know already for a very long time that 1602 indicates the presence of ergosterols in lipids and lipid droplets. And 1004 is technically protein signatures coming from filanelin bands. So you can now generate images. All we do is, take one Raman spectrum at each point like that and keep moving to different points. Each pixel that you see here is coming from, that means that we have measured one spectrum in that point, right? So that means several hundred spectra, probably 600 or 700 spectra for each cell we measure, and then we can reconstruct all these, beautiful molecular images. However, please be sure that we do not use anything else. We just take the cell and shine the laser on. That's it, and which is also very low power. It doesn't kill. It doesn't, break the cell, and there are no labels. There are no fluorescent labels. Nothing, right? So all the colors that you see are these false colors, which we just, apply. But these are molecular images. So we can call it as label-free molecular imaging, right? And you see, in the case of spores, because spores are very quiescent forms which have very low activity, so we can see protein metabolic activity is very low compared to the vegetative cells which are actively dividing. And also, in the case of spores, they are trying to store more lipids compared to normal cells. However, the more interesting part is that the beta glucans. So beta glucans, of course, are known to be in the cell wall. Of course, that's fine. We do see that in the normal cells and also in the case of spores. However, we fail to see the presence of beta glucan in the newly formed spore walls. It's only in the mother wall because this is the result of two individual cells coming together. But if you see, you can see these four round circles inside, which are actually indicative of alpha glucans in the spores, right? It's so surprising because if you take colocalization images of both alpha and beta, means only where both alpha and beta are present, it will show some information or signal. Then it shows that only only in the mother cell wall, both components are present, but in spore walls, only alpha is present, which was not known, right? So now further we have obtained some sort of label-free alpha and beta images, glucan images, but we also have more components such as mannan, especially because that's also one major component of the cell wall, which we cannot identify with only single band. So for that, what we do, we apply something called as multivariate curve resolution analysis, where we can extract pure molecular information from that large data set, where, for example, we of course try several models. It's not very straightforward. It takes some time to master it, but once you understand how to do that, it is very, very powerful in order to extract pure molecular components. And if you see, the first 5 components, these are modeled spectra, right? These are not obtained Raman spectra, but these are modeled based on the real data. And we obtain again proteins and lipids, but we could also extract 2 independent saccharide components, which is now showing that one of the saccharide component 1 is beta glucan, but general glucans, but saccharide 2 is actually mannan. That means we are able to get extra information, and then we can also construct similar corresponding molecular images. So now these are not the same images because the previous images were obtained from one single band, but this set of images were obtained from the whole spectra, right? These are totally different, and then of course the contrast is much better in terms of the known information of lipids and proteins. But all you can interesting point that we can see is the presence of mannan. The mannan is actually not only present in the spore wall but also present in the spores. So such a presence of mannan is very, very clear, especially if you look at the colocalization of both glucan and mannan images. So that means for the first time we have identified structurally very similar components, 3 independent components, in a label-free manner, and that gives us a much more biochemical biomolecular information to understand how mitosis or meiosis actually happens. Well, how can we actually use it? So what? Okay, you understand this single cells, you understand this. So what? This is especially useful because these are fungal models, right? And especially in the case of pharmaceutical industry, people are always trying to make some new drugs, new antifungal drugs, right? So if we, and also these antifungal drugs are primarily targeting these cell walls, right? In order to break them up. So, for example, we can test the efficacy of various novel compounds for antifungal activity in the single label-free manner of single cells, right? And also we can try and use this information to make diagnostic, applications. For example, in the case of fungal infections, probably we can try to, detect the presence of such cell walls in the blood or, in serum and so on, where we can really make use of it very well, right? So I think I'm going very slow. I just have only one. So how much more time do I have? So this is
Sir, you can expedite as soon as you can. No problem at all.
okay, because there are some very interesting results that I would be very happy
Yes,
to
sir.
share.
Yes, sir. You can show the results. No problem at all.
Okay. So, so that's how we can actually take this fundamental information and develop some new applications. Well, some of the results have already come in, and I'm going to show you the imaging results for the drug testing analysis. Where, for example, if you take a drug that is terbinafine, terbinafine is going to stop the synthesis of ergosterol by inhibiting squalene epoxidase, which then stops the conversion of squalene to epoxysqualene. This is one of the major metabolic pathways, steps involved in ergosterol biosynthesis. That means if you stop this reaction, this ergosterol cannot form, and they cannot form good cell walls, and then eventually we see the antifungal activity. So I think I already mentioned to that. So there are several steps in between, squalene epoxidase, epoxide, and ergosterol. So none of these will happen, so it cannot form functional ergosterol. So you see, we have already observed lipid droplets inside the cells that contain primarily ergosterol, and the signature is coming, the 1602 it tells us about the presence of ergosterol. And if the drug functions well, then we see the accumulation of squalene, which is usually not observed in yeast cells, because usually squalene will move and be converted into epoxysqualene. And also, this is a simple, straightforward, Raman spectrum of the drug, where we have some sort of a triple bond here, which is showing up as 2219, right? So we can use any of these strong bands as a marker for the drug itself. So that means, for example, if you just take the cell to the platform and you add the drug into the medium, and then immediately within 15 minutes, right? This is 20-minute spectrum, right? So you follow the same cell, same point, you just add the drug into the medium, and after some time, you are able to actually see the presence of drug. You see, there are several strong bands that are indicative of the drug inside the cells. So if you do imaging, for example, similarly, for example, this is before addition, and this is after addition, roughly about 20 minutes, you can see this is the drug. And that means direct drug imaging is possible when you're using Raman microscopy. Well, that's wonderful, because that's exactly what pharmaceutical industry wants. You can look at the drug inside living cells, where they are and whether they are working well or not. Well, that we can check more in detail by doing spectroscopy, where you can follow the time resolved information at different times. You follow the cells, and then the Raman spectrum is going to give you very detailed information about what is changing at the metabolic molecular level. For example, you can see the presence of drug up to about 8 hours, and then it's gone again, and so many things are happening, right? You can actually do very detailed analysis. For the sake of time, let me not spend too much on the details, but must understand that we are able to extract a lot of information about the metabolic activity, not only for the lipids, but also for many other components. But it's always better to see. Seeing is kind of believing for many people. So of course, for scientists, it's very interesting to actually take images. So again, we can do molecular imaging. Again, you see this is drug, these lipids generally. So the lipids are increasing around 8 hours and so on. But to begin with, at 0 hours, there is no drug. But half an hour later, you can see very strong presence of drug inside the cell. And then
Sir,
yes.
excuse me, please, sir, if you can expedite it now, because we are having few papers as well.
Oh yeah, I'm finishing it now. So
No
this
problem at all. Yes, sir.
so Yeah, so we can do this kind of drug imaging and also look at detailed molecular analysis. So I'll okay, diagnostics. I think I'll skip. I'll keep it for next time. And then thank you so much for the opportunity. Thank you.
Thank you very much, Dr. Hemant, for highlighting the particular points that is related to rheumatoscopy. However, we can understand it is not easy to explain such points in a very short time because the spectroscopy itself explanation is very high. So for this, we just try to incorporate because I also work in the Raman spectroscopy as a
Oh,
physicist, and we just try to carry out this work in the particular lab. So that is a
okay,
synthesis and application part. So I
okay.
am happy to see your work. So that is the results and discussion are very nice. And the presentation in terms of doping and other material for the application point of view, that is also excellent. Thank you very
Oh,
much. And my extreme
thank you.
thanks to you, sir. Thank you.
Thank you. Anytime.
Thank you so much, sir, for such an enlightening presentation on application of AI-assisted Raman microscopy. Your work in advancing diagnostic method through AI and Raman spectroscopy provides incredible insights to all of us, and we deeply appreciate your time, expertise, and contribution to this conference. Thank you once again, sir.
Thank you very much.
Now we are going to move to few of the topic of presentation by the research scholars. Pankaj sir, can you please go through the research scholars which I have sent you to through the evaluation sheet?
Yes, yes, I'm just opening the sheet. First one is a Vikrant Chaudhary. Significance of forest ecosystem towards maintaining ecological sustainability. I think he's not there in the
Yes
meeting.
sir, yes, we can skip that one.
Second was is Vinay Vikram Singh. I think the particular those who are present here on the basis of this we can start. If anyone is there, then he will say.
Yes sir, we can skip the Vinay. I think you another one is Vaishnavi A Nair,
Yes, Vaishnavi and Iyer is there.
CP50.
Yes, CP50.
CP50?
Yes,
Yes, we
Vaishnavi
can.
CP50.
Vaishnavi, do you want to share your presentation right now?
Yes, ma'am.
Okay, now you can share it.
Okay.
Dr. Nidhi, I request you just brief about the timing so that accordingly she will finish the time.
Yes, you have only 7 minutes for your presentation. So
Okay.
kindly expedite it within that one.
Okay, ma'am. Okay, ma'am. Can you see the slide?
Yes, your slide is clearly visible. You can start your presentation now.
Okay, ma'am. Thank you. So, good afternoon, everyone. I am Vaishnavi Nair from Integrated Science, fifth sem student. I am here to present the utilization of biochar for wastewater, removal of heavy metals
Vaishnavi,
from
your voice is
wastewater.
cracking now. Vaishnavi,
Pardon, sir?
your voice is cracking.
Sir, can
Anyway,
you hear me
just
right now?
speak louder and start, no issue. Hai
Sir, can you hear my voice right now?
Anyway, you just start, no issue. Perfectly all right. We start. Hai na?
Oh, thank you. So I am here to share you the topic about the utilization of biochar for heavy metal removal from wastewater. So first, what is heavy metals? Heavy metals are generally considered those whose density exceeds 5 gram per cubic centimeter. So most of the elements falls into this category are highly water soluble. And according to the toxicity, they are well-known toxics and carcinogenic agents. Some of the common heavy metals are copper, silver, zinc, cadmium, gold, mercury, and so on. So now sources of heavy metal into the environment. How heavy metals can be found in the environment? There are various ways, such as industrial discharges, runoff, leaching, agricultural practices, atmospheric deposition, natural sources. Next, the impacts of heavy metal. How heavy metal impacts us? It can impact us through various ways, through destruction of aquatic ecosystems, reduced biodiversity, physiological and behavioral changes, bioaccumulation, which led to biomagnification, human health risks, and contaminated soil, groundwater, and so on. So now, biochar. What is biochar? Biochar is a kind of carbon-rich porous substance with abundant active organic functional groups and carbon aromatic structure with a neutral to alkaline pH value, relatively high cationic exchange capacity, large specific surface area. So large specific surface area, which means it has large absorption capacity and negative surface charge. So now we are going through the preparation of biochar. How biochar is prepared. So in this figure, you can see that biochar is prepared through various methods like flash carbonization, gasification, pyrolysis, hydrothermal carbonization, torrefaction. Torrefaction, which means simply means mild pyrolysis. So first we will discuss about pyrolysis. Pyrolysis. Pyrolysis is the thermal decomposition of organic matter in the absence of oxygen. So the temperature can be ranged between 300 to 700 degrees Celsius, and it produces carbon-rich biochar. And gasification. In gasification, the biomass is converted to syngas, that is a mixture of hydrogen and carbon monoxide to partial oxidation at higher temperatures. This is, we can say that it's around above 700 degrees Celsius. And the remaining solid part is for the biochar. Hydrothermal carbonization, or HTC. It is the treatment of biomass with water at elevated temperatures and pressures. It is effective for wet feedstocks like sewage sludge. Flash carbonization or rapid pyrolysis is at the high is at high temperatures and low residence time. It produces biochar quickly. Impacts of heavy metals. There are certain impacts of heavy metals. Heavy metals. Types of biochar. So there are certain types of biochar, like feedstocks-based types biochar, preparation method-based types, and functional types. So here, agricultural residue biochar is the most common biochar that we can see. It is straw, corn stover, and rices. Similarly, others. Now, how biochar can absorb heavy metals from the environment, such as soil and water. So in this figure, we can see there are certain mechanisms shown here, such as surface compressation, electrostatic force, reduction, co-precipitation. So in surface compressation, biochar can be found in the biochar surface contains functional groups like carboxyl and hydroxyl groups. And this group can attract and bind heavy metals through chemical reactions. Now, electrostatic force. What is electrostatic force? Electrostatic force is the attraction between auxiliary charged ions. So biochar develops surface charge, which can be either negative or positive, based on solution pH. Reduction. So biochar can act as a reducing agent. It converts higher oxidation state of heavy metals to lower oxidation state. So for an example, for chromium 6 to chromium 3. For precipitation, formation of insoluble compounds with heavy metals. It facilitates precipitation and removal. Objective. Objective is to determine the efficiency of biochar in removing various heavy metals. Second one, to identify the most effective type of biochar for heavy metal remediation. Methodology. A comprehensive literature review was conducted using research articles sourced from Google Scholar. Relevant keywords included heavy metals, wastewater, absorption, and biochar. A total of 40 peer-reviewed research articles were selected for in-depth analysis to inform the review results. So, as we can see the pie chart, we have a diverse range of feedstocks contributing to the biochar production. Here we can see wheat straw and rice hulls are the prominent biochar we used, followed by coniferous shells, black bush waste, sawdust, sugarcane baggage, coconut husk, so on. So next one is the absorbed amount of heavy metals by biochar. Here we took many heavy metals so to arrange according to the absorption capacities. The first one, absorbed amount of cadmium. So we can see that in the first one, the cadmium, which is having the maximum absorption capacity, which is around 649.9, shows a maximum absorption capacity, and family ultra biochar is used in this case. And in the lowest case, that is around 46.9, here constant biochar is used. And the next one is the absorbed amount of lead. For lead,
Maybe your time is
which
over now.
is
So wind up within 1 minute, okay?
okay, sir. So in lead, we used around 384.08. The absorption capacity was 300, more than 300 milligrams per gram. So here we use the Procter wheat biochar, and in the arsenic, for arsenic, it is 104.55 milligrams per gram, and we use the scallion sheetings biochar. And for the mercury, 13.55, we used the sugarcane baggage biochar. And for copper, the maximum absorption capacity is shown here is 17.01. We use sawdust biochar for this, and the absorbed amount of zinc, which is shown here, is 35.75, rice husk is used as biochar. Now, for future perspectives, we can for optimization of biochar production, field application studies, environmental and economic assessment, policy and regulatory support. Conclusion. So we can conclude that biochar is an effective absorbent for heavy metals. The efficiency depends on many factors such as feedstock, surface properties of the biochar, the temperature, etc. And among the different types of biochar we analyzed, some for specific heavy metals, they have superior absorption capacities. So, so some heavy metals are more suitable for absorption. So this review highlights the potential of biochar as an economical and environmentally sustainable solution for heavy metal removal in wastewater treatment, offering insights into optimizing biochar properties for improved remediation and efficiency. Thank you, sir.
Okay. Thank you very much, Vaishnavi. Can you highlight one application that is related for the society that is beneficial in terms of biochar and others?
Pardon?
You just highlight any one application that impact for the society, especially that is work carried out by you.
Yes, sir. Industrial wastewater, we can so heavy metals are produced in these water bodies. So using any biochar, we can actually reduce the toxicity of heavy metals present in the water. So it will be benefits to marine life, even human health.
Okay, can you give the name of any one heavy metal that is mostly used in this particular process?
Cadmium. Cadmium is mostly
Cadmium.
used. Arsenic is mostly used.
Okay, okay. Okay, thank you very much for nice presentation. Thank you very much.
Thank you, sir. Thank you, sir.
Next one is Mr. Mangalam is there. Mangalam, Sony and Sonal Singh. Anyone is there?
Yes, sir.
You just share your presentation and try to wind up within the time frame.
Yes, sir. Yes, sir. So, good afternoon, good evening to all. Myself, Mangalam Soni. Here I am going to present my research, a review type paper. It's on one.
Mangalam, you just show in a F5 mode slide.
Yes, sir. Yes, I'm doing. So my topic is in phytoremediation. Its application is to prevent post-toxic effect due to hydrocarbon spillage. So first of all, what is phytoremediation? Phytoremediation is a bioremediation process that used to plant to clean up soil, air, water, and contaminate with the hazardous pollutants, including hydrocarbon from oil spill. So after oil spill, it has been observed that the many toxic effects are observed in environment, soil and water. So we can reduce by the help of plant which incorporate them into a physiological process, or it can bind to them themselves. So so that the contamination or the heavy metal or something else that affect the environment can be decreased up to certain limit. So certain plants can absorb, accumulate, sometimes even transfer these toxic substances, making them useful for environmental cleanups. It is particularly effective for treating hydrocarbon spillage, offering a sustainable and cost-effective solution. So as compared to chemical and physical method, we can use this biological method, and it is like cost-effective even. So carbohydrates pollutant arise from oil spill, industry discharge, and petroleum lakes. These toxic compounds are severely impact ecosystem, leading to loss of biodiversity and health hazards for marine ecosystem and human. Understanding the source of effect of hydrocarbon pollution is essential for effective remediation strategies. Even though we through transportation or from any industry, the spillage will occur, and even though it can affect environment, so we can remediate the toxic effect of spillage. So phytoremediation employs several mechanisms to combat hydrocarbon toxicity, including phytodegradation, where plant breakdown contaminates, phytosterilization, where plant breakdown contaminates, and immobilize pollutant in the soil. And the last is these processes enhance the natural degradation of hydrocarbon, making it viable option for remediation efforts. So these are the some plant are listed here. So here we use alfalfa, that is Medico sativa, that help to stabilize the soil and support microbial community for degradation. Next is willow and poplar, and which has been used to absorb carbohydrate from deeper soil layer. And next is brassica, that is we use in sarso, we used to say in common language, effective for heavy metal absorbent from hydrocarbon contaminated site. These are the well-known example of which through which we can reduce the amount of contamination where the spillage has been occurred. These are the benefits of phytoremediation instead of using physical or chemical method. Phytoremediation offers numerous advantages, including cost-effective, environment sustainable, even it is environmental friendly also. It does not affect much as compared to chemical method, and aesthetic movement and improvement of contaminated sites. It is a natural process that enhances soil health, promote biodiversity, making a preferred method for environmental restoration. So we can restore the environmental condition which has been affected by the spillage, so can be restored by the phytoremediation. And these are some case studies have been reported in Sweden and US. So in the a former gas station is in Sweden was contaminated with petroleum hydrocarbon posing a threat to nearby ground. So they have used willow tree, which has been used to stabilize or hold the contaminants within the soil. And second is used in US. They use brassica to absorb the heavy metal due to the spillage in the soil. And despite some disadvantage has also been observed. Despite its benefit, Phytoremediation faces challenges such as slow remediation, potential plant toxicity, and that need to be site-specific assessment. According to site-specific assessment or spillage, we have to plant the tree so it can do as function according to their source of contamination. Understanding these limitations is crucial for optimizing phytoremediation strategies and ensuring successful outcomes. So here I will conclude all overall. Phytoremediation represents promising approach to tackle hydrocarbon spillage toxicity. As a result, advance and technology improves. The integration of genetic modified plant and biostimulants may enhance its efficiency. Embracing this natural process is vital for sustainable environmental management. So we can use biological method instead of chemical or physical method. So it is, and even it is biosustainable and environmental friendly method. Yes.
Okay, Mangalam, can you highlight any two limitations of this work? So that is.
So limitation is that during if some spillage has been occur and we have to plant the tree according to their conditions. We cannot plant the tree whichever we need. So we have to just analyze, and we can only incorporate or plant that tree which help to reduce the contamination.
Okay, in this case, can you suggest any one application that is benefit for the
Sir,
society?
application where the around the industry of petroleum, we can plant the brassica. That is we used to call sarson. So if in case any spillage occur, or so it can absorb the heavy metal and it can prevent contamination other water bodies or in environment.
Okay, thank you very much for.
Yes.
Okay, thank you. So just I'm calling Vinay Vikram, Akash Gupta, Manish Kumar, anyone? Siddharth Sharma, Rajiv Goyal, Neha Sharma, Nandan, Mohini Dagar, Megha, Madhurima, Kuldeep Soni. Kuldeep Soni is here.
Very much, sir, for your talk. We have
Kuldeep Soni is here.
factors regarding the management, service management, and
Is Kuldeep here?
so I hope that people will be
Not
getting
there in this particular participant list.
Okay,
for
okay, okay.
future opportunities.
Rajiv Goyal,
That will help.
No,
Vinay Vikram.
So, okay, so we have come to the end
One
of
is
the
also not there, sir.
now we'll be starting the
Vinay
voting
Vikram not
session.
here. Okay.
Yes,
Only
sir.
Sonal is visible in this list. Sonal Singh,
Yeah,
others
and
are
we'll be in the
part of the
voting
team.
as we will be starting the voting session.
Hemant.
I will be handing over to
Dr. Seema Singh.
to conduct the
Chandra,
voting session. Just give us a minute, we will be starting.
Okay, only few. Sir, sir. बच्चे तो है नहीं
Anyone
इसमें
is there in the participant who have not presented
बच्चे
the.
तो है ही नहीं इसमें जिसको देना है सब मैसेज किया था
I think Dr. Abhishek, no one is there.
sir wait we are calling those students who not joined here.
Dr. Seema Singh is written now. Seema Singh is present.
Okay.
I'm present, sir.
Okay, do you want to give your presentation here?
Yes, sir.
Okay.
Very shortly, I will complete my presentation, sir.
Okay, start please.
Here is
Okay, sir.
Start.
Sir, 1 minute.
NCTS.
Sir, are you able to see my presentation?
Yes, yes. Yes, you are audible here. Start.
Okay, sir. Very warm good evening to all the members present here. I'm going to give my presentation on the study on zooplankton diversity in a freshwater pond Rajabandh of Jamtara. Also, I have done this kind of work from so many ponds to study, to check the water pollution level in the different water bodies. And I will summarize my presentation in very shortly, sir. Zooplanktons are the cosmopolitan in nature. As we know, it is the most important ecological parameter in the water quality assessment because they help in the disposal of the sewage and act as a natural purifier of water. And present study deal with the zooplankton diversity throughout the year in the Rajabandh of Jamtara, Jharkhand. Or various type of zooplanktons was studied, and this study revealed that the zooplanktons reported mostly were the Rotifera, Cladocera, Copopoda, and the Ostracoda. And as we know that they are the central member to manage the different trophic level of the water, as actually fishes feed them, and they also help in cleaning of water body, as well as it is a good indicator of the understanding the water pollution status. So I will, during my research work, actually I have done my research work by using the different, very simple method of Bettish and using the Sedgwick rafter counting chamber. And then I found that in the Jharkhand, different pond, actually we observed, different water body, we observed that the 14 different kind of zooplankton were observed. Out of them, the 4 groups are predominant. They were the Rotifera, Cladocera, Copepoda, and the Ostracoda. And all the 4 group, maximum abundance of the Rotifera were found up to the 38%. But when we have studies during the rainy season and the area nearby where the local people uses the water body, we observe that the abundance of the zooplankton reduced. So my studies focused how the water pollution affect the population of zooplankton and how we can reduce the water pollution in the water body, and how we can use zooplankton as a tool, as an indicator for the water pollution. This is the main theme of my study, and actually my study is very large, but I have summarized in a very small presentation PPT. So now this is the summary of my research work, and now thank you, sir.
Okay, thank you very much. Can you highlight any one point that is related to the density order observed by you in a particular reflector?
Yes, I have observed that the Rotifera were found mostly up to the 38%. And we found also that I have studied in 3 to 4 ponds in the different places of the Jharkhand, and such as the Shivganga of Deoghar Bajnath Dham Shivganga pond is actually during the actually Preship Sawan Mela, it got polluted and the population of zooplankton reduces. So that was the main theme why I have started my work. And mostly population of rotifers were found in the different ponds of 4 different places, sir. And it can be a best indicator of the water pollution. This is the main theme and main focus area of my research, sir.
Okay, which species you found better in terms of this particular task?
What, sir? What, sir?
Species that you carried out, 5 species for this particular, 4 species for this demonstration. That observation taken by you using the different species in which
Yes,
you
sir.
found.
Yes, sir. No, sir. I'm actually your voice is disconnected. That's why I'm asking you again, sir. Please.
Okay, I wish to know about the particular work that is carried out by you in terms of the different species.
Okay,
So
sir.
which you found
Sir,
better in terms of.
yes, sir. Sir, they are the amnia, Sclipocera, such kind of species were observed during this study, sir.
Okay, okay. Thank you very much. The nice presentation.
Thank you, sir. Thank you.
Okay, thank you, Seema. So, anyone going to present here their paper?
Anyone is
waiting.
there in the participant who are not presented now? Anyone left? I think
Participants?
no one is there. The Dr. Pita.
Sir, wait, sir, wait.
One is joined now.
Ekans, are you here? Ekans. Anyone? Anyone not given, please give presentation. कंप्यूटर से कर ओके डालिए सर नहीं आ रहा है अच्छा Okay, P. Shekhar, am I audible? P. Shekhar.
You are audible, sir.
Shekhar.
P. Shekhar is
Please
not.
give
Is it
your
audible to you?
Yes, you are audible.
Yeah, please.
Yes, please give your presentation.
Yes, yeah. Okay, sir. Hello.
Yes, you may start your presentation.
Yes, yes, yeah. So today's my presentation is regarding bioreactors as a sustainable energy houses, innovations and applications of carbon neutral energy production. So I am Dr. Peshekar, working as an associate dean and professor. So this is my outline of the presentation, introduction and purpose of the presentation, and types of bioreactors in energy production, and innovations in bioreactor technology for carbon neutral energy, and applications and case studies of bioreactors, challenges, and call to action. So if you see the introduction and purpose here, our in the main major context of climate change and carbon emissions, as the world grapples with the impacts of climate change, reducing greenhouse gas emissions has become global priority. As Intergovernmental Panel on Climate Change, IPCC, has made it clear to avoid, to avoid the most severe impacts of climate change, we must drastically cut carbon emissions, aiming for carbon neutrality by mid-century. So in this challenges in the energy sector, while renewable energy sources like solar, wind, and hydropower offer some solutions, they have infested with some challenges such as energy storage, resource availability, and grid integration create the need for complementary technologies. So in this particular thing, purpose of the presentation is the bioreactors' role in sustainable energy. So these bioreactors can be called as a sustainable energy houses, then can produce renewable energy while addressing the waste management and emission reduction. So in this particular presentation, we aim to showcase how they can serve as a practical decentralized energy sources and bridging the gap in the current energy landscape. This actually the adaptation of these bioreactors for waste to energy conversion, ability to capture carbon, and potential for localized energy production make them valuable tools for rural, urban, and industrial applications. So significance of the bioreactors, if you see here, bioreactors represent a complementary approach that can transform waste materials into energy and sequester carbon, closing loops in the carbon cycle. So bioreactors offer a flexible energy solution, which is particularly important for energy resilience in rural communities, remote areas, where grid access is limited. So these bioreactors can empower communities to generate their own clean energy. So key takeaways, as we just I'm moving, the types of bioreactors for carbon neutral energy production. There are different types of bioreactors are there. One is first algal bioreactor. So algal bioreactors use microalgae, which can photosynthesize to convert CO2 and sunlight into biomass. So these algal bioreactors can capture carbon dioxide from industrial emissions, making them useful for carbon capture and utilization. This is a very useful application of algal bioreactors. And this is the picture of algal bioreactors where you can use it in an industrial purpose for capturing of the carbon dioxide. Next one is microbial fuel cells. Especially these are used for generating of electricity directly from the organic matter. And these reaction also produces a byproduct of clean water, making MFCs beneficial for wastewater treatment. So the end product of this MFC is nothing but clean water. And this is the mechanism of how this MFCs work. And photosynthetic bioreactors utilize photosynthetic microorganisms such as cyanobacteria and specific algae to convert sunlight and CO2 into biofuels. This process mimics natural photosynthesis, enabling carbon fixation and biofuel production without relying on the organic waste. And these are some of the advanced photosynthetic bioreactors. These are called as the tube bioreactors, and these are called flat bioreactors. And the next one is bioelectrochemical system, which can be called as a MES. They are especially useful in converting organic matter into hydrogen gas. So this aids in hydrogen production, which can be collected as a clean energy source. So this particular MES plays a very important role in eco-friendly environment. That is, they produce hydrogen gas, which can be used as a clean energy. And this is they are using some external power supply. Hydrogen can be produced. And next one is hybrid bioreactors. what is the meaning of hybrid bioreactors is they will combine one or more renewable energy sources like solar and geothermal to capture the carbon. So, for example, a solar-powered algal bioreactor can harness sunlight to boost algal growth and CO2 capture efficiency. So, this is the one example of this particular hybrid bioreactor. Here, these are the solar panels and these are the algal bioreactors. By combining these two, we can capture the carbon dioxide. So applications and case studies of bioreactors here. So what is the application of microbial fuel cells? Especially they are useful in wastewater treatment, remote and off-grid energy. And these microbial fuel cells are very, very important where there is no grid electricity generation, like remote areas. And third one is microbial fuel cells are helping carbon neutrality. Example, Thames Water in UK, they're using MFCs to produce electricity generation from the wastewaters. Second one is carbon capture. This one is the algal bioreactors. They are helping carbon capture and sequestration, that is called CCS. And second one is for biofuel production. Third one is sustainable agriculture. So all these sectors, these bioreactors have their applications. Example, Alga Energy in Spain has developed photobioreactors that capture CO2 from industrial sources and convert into biofuels. Next one is photosynthetic bioreactors, applications in solar energy capture, energy production, as well as the enhanced carbon sequestration. So these bioreactors act as a natural carbon sinks. These photosynthetic bioreactors can act as a natural carbon sinks, contributing to climate change mitigation efforts. Example, in IHI Corporation, Japan, they have developed large-scale photosynthetic bioreactors that can capture CO2 from the industrial emissions. Last one is the bioelectric chemical system, BES, and they have applications in hydrogen production. Especially this BES is mainly used for hydrogen production, which is a very clean energy source. And they can be used for wastewater treatment as well as the renewable energy source. Especially at IIT Delhi in India, they are working in bioelectrical systems that convert organic waste into hydrogen. It is in the pilot
Please
laboratory
conclude,
stage.
sir. Please
Yes,
conclude.
yeah. Hybrid bioreactors application is integrated waste to energy, synergetic in carbon capture and sustainable local energy solutions. So here, if you see, I'm going. Challenges in bioreactor technology, mainly infrastructure and technological costs, financial returns and market demand, complexity of biological process, because all these bioreactors mainly depend on the biological organisms. So these processes are very complex in nature and maintaining is somewhat difficult. Scalability issues, especially from lab to pilot to full industrial scale. And third one is the maintenance and reliability. And resource availability also another constraint for this bioreactor application for energy sources. One is feedstock supply, water and land use, and energy input requirements, and limited carbon capture efficiency. When compared to the normal renewable energy sources, these bioreactors have a limited carbon capture efficiency, even though they are very efficient, and storage and use of captured CO2, and regulatory and policy barriers, and the conclusions call to action, especially for accelerating the adoption of bioreactor technology. There should be some innovation in bioreactor designers, multidisciplinary collaboration, government incentives for clean energy technologies, and carbon pricing and market mechanisms. So all these comes under government policies by which we can encourage the production of these bioreactors for clean energy, and scaling up of deployment and sustainability across value chain, especially circular economy approach. The full potential of bioreactors will be realized when they are integrated into circular economy model and life cycle analysis, and policy awareness, creating the education and community-based initiatives. So all this comes under this bioreactor applications in clean energy. Thank you very much.
Okay, thank you very much. Will you signify your work? What is your contribution in this work? Or what is the outcome of your work?
Yes, sir. Actually, I am working I have produced one renewable energy biomass briquettes which using this bioreactors.
Okay, which bioreactor you found most suitable for this particular society?
Yes, sir.
Which bioreactor you found is most suitable, especially for the specific application?
So actually, it is in the pilot scale. Actually, we are trying now.
Perfect. Okay, thank you very much.
Yeah.
Yes, Dr. Abhishek, over from my side.
Okay, thank you. Thank you for your presentation. Now we call to the Ekans or Nandan. Please start your presentation. Either Ekans or Nandan. Ekans or Nandan. Am I audible?
Yes, sir. Am I audible, sir?
Yes, please start.
Am I visible, sir?
Yes, visible.
Okay. Good afternoon, everyone. I am Ekansh Agarwal and presenting my topic on optimization greenhouse
Yes,
design.
you can start.
Little louder, Akash, little louder.
I am Ekansh Agarwal and I am presenting my topic on optimizing greenhouse design for sustainable growth and efficiency. So, greenhouse gas. What is greenhouse gas? Greenhouse gases are those atmospheric gases that are responsible to maintain the Earth temperature. Otherwise, the temperature would be around -18 to -20 degrees Celsius, which will be responsible in which the major greenhouse gases which are responsible for that are carbon dioxide, methane, nitrous oxide, water vapor, ozone, and fluorinated gases, and other all types of gases. And global warming potential, that is GWP, the effectiveness and of different greenhouse gases contributing to global warming is measured using global warming potential. For instance, methane has a GWP of 81.2 over 20 years. Human impact, in the case of human impact, we can see the industrial revolution. Since industrial revolution, human activities have dramatically increased the concentration of greenhouse gases in the atmosphere. So then what the humans are doing, human activities in contributing in greenhouse emissions like fossil fuel combustion, transportation, industrial processes, and many more. Like we can see the EV cars and bikes are running on roads nowadays, but the charging facilities which are available, is the charging stations are allotted, and even at their house they manage to charge. But the charging station, when and houses, when they went out from electricity, then they use generators, and those generators are automatically they use diesels or fuels, which ultimately release the gas which is harmful for the environment. So we have to minimize this to save our environment. In this order, we can construct organic solar cells. Then what is organic solar cells? These are the type of photovoltaic technology made of carbon-based materials. Unlike traditional silicon panels, that they are semi-transparent, lightweight, and can be tuned to specific wavelength. Organic solar cells are significantly enhance the greenhouse environment by integrating renewable energy generation with optimized condition for plant growth, for contributing more sustainable agriculture systems. What are the key ways? The key ways are energy demand reduction, wavelength selectivity.
Please conclude. Please conclude. And
Yes,
who
sir.
is giving next? Please tell me.
So in that case, the
Raise
climate
the hand. Who one is the next?
mitigation in climate change is more important, and it will mitigate the climate change. And this is the organic solar cell construction, in which we can construct like this, and it will be help in many applications, like we can use to charge our mobile cells and batteries.
Is Nandan here?
Also,
Raise the hand.
we can conclude that we have to use the 3 R's which are responsible: reduce, reuse, and recycle. Thank you, and save the environment.
Okay, thank you, Ekans, for your good presentation.
Thank you, sir.
Who is next now?
Thank you. Next one, please. Next one, please.
Anyone left who are present in the particular participant list?
Yes, sir, I am.
May I start, sir?
Yes, please start.
Okay, sir. Let me
Harika, you can start.
Okay, sir. Good afternoon, sir. I'm Niharika Singh, and today I'm going to present my topic on transforming waste into value-added products, a sustainable approach. So there are gases like carbon dioxide, ozone, and methane and nitrous oxide that trap heat to maintain the earth temperature. But the human-induced waste and emission become a major cause as it allows the global warming and climatic change and other environmental stress. This raised concern needs to be addressed by sustainable practices and technologies to protect our environment. So the strategy is the transforming waste into value-added products to promote sustainability and resource efficiency. This approach involves the transformation of waste into valuable products like fertilizers, chemicals, bioplastic, etc. And it focuses on the valorization, that is recycling or reusing the waste where discarded materials are converted into valuable resources to contribute to our circular economy. These are some examples that from waste to value-added products, such as sustainable body care products. Like we can use coffee grounds to make soaps, scrubs, and other body oils, etc. Leveraging their natural antioxidant and exfoliating properties. Another one is nutraceuticals. Like food waste can be processed to extract bioactive compounds like vitamins, proteins, and we can use it in dietary supplements, enhancing our health benefits. Another one is bioplastics, biofuels, and a process like pyrolysis where the heating plastic waste in the absence of oxygen can convert it into oils, which can be refined into fuels or chemicals. So we are focusing on the coffee waste. So we are focusing on the coffee waste where from coffee we can make sustainable body care products, raw fertilizer and odor control spray where we can reduce the unwanted smell. We can use the bioplastic like cutlery and all from coffee beans. And also we can use it in furniture touch-ups like oils, creams, ointments to furnish the furniture. And so waste plays the major role in environmental stress like causing pollution, gas emission, climate change, and even death of living beings. So basic practices like we can do in our natural life, daily regular life, like so here I am presenting like we can reduce the plastic uses, we can do carpooling, plant trees or uses of sustainable product that leads to waste reduction, low gas emission and less pollution, and which will further promote sustainability and prevent environmental stress. So at institute level and students, we can do things to promote environmental sustainability and reduce waste through like colleges can eliminate single-use plastic water bottles and soda bottles during facilities like dining and vending machines, or we can bring our own cup for coffee, tea to reduce use of plastic and paper disposable cups. We can use digital textbook and resources, even like several printout and hard copy should be avoided to manage like paper waste, or we can opt for recycled paper or tree-free papers that we can use from, like we can use rubber paper that can help in waste reduction. Also, we can utilize usable cloth, towels, and napkins instead of disposable ones. Also, students can arrange like management programs, environmental clubs, and promote recycling and participate in composting. This is the conclusion. So we can achieve sustainable future by like converting waste into valuable products through various processes like pyrolysis, anaerobic digestion, biogas plants, and this waste can be converted into energy reducing that will reduce greenhouse emission gases and promote a circular economy. This sustainable approach not only mitigates waste management issues but also generates new revenue streams and job opportunities. By embracing this approach, a more sustainable future can be created, minimizing waste environmental impact while
Mohini,
maximizing.
am I audible?
Yes, sir.
Please ready for the next presentation.
Thank you, sir.
Okay, sir.
So let's convert trash into treasure. Thank you, sir.
Okay, thank you very much.
Okay, start your presentation, please, Mohini.
Mohini, you just summarize your work in terms of your outcome, what you did and what is the outcome and application part in this particular presentation.
Yes, sir.
Please.
Sir, actually, I'm presenting a review of constructed wetland. Over the past few years, pharmaceutical has grown significantly. Various classes of pharmaceuticals are found widely in the water bodies at trace level. According to a study of Femina Caroline, approx 12,000 active chemicals were identified, out of which 713 were found in the wastewater. Pharmaceuticals have high toxicity. They are more persistent, bioaccumulated, and easily enter into our food chain. So, sir, there is concern to for the wastewater treatment. Treatment of wastewater is a challenge for the researcher because a lot of money is invested in wastewater process. It also does not ensure the complete removal of the pollutant, and a lot of energy is also required. Hence, a treatment with eco-friendly approach is needed. Constructed wetlands seen as alternative method for the wastewater treatment. Constructed wetlands with the help of plant root, substrate material, it remove pollutant by naturally breakdown, help in filtering the water by physical and biological process. It is energy saving, environment friendly, low cost, low energy consumption. So the synergic effect of plant filling material and microbes effective treat the wastewater. Filling material in the constructive wetland play an important role. Biochar as a filling material can be a better option. Constructed wetland with biochar has emerged as an innovative approach for pharmaceutical treatment. Biochar has some specific property which make it suitable candidate to enhance the performance of constructed wetland. It is carbon-rich product, large surface area, high porous structure, high cation exchange capacity, abundant functional group. After reviewing the last year paper, I can see the positive result of biochar embedded constructed wetland for the removal of pharmaceutical. In this poster, in this figure, you can see I made a constructed wetland with the different three layer. With the help of reviewing many paper, I'm thinking about I'm thinking to continue on this topic. Thank you, sir.
Okay, thank you. Kindly tell me about your significance. And next one is the please start. Seema. Is Seema here? Please start. Who is not given presentation?
Anyone left who have not given the presentation?
Oh, sir, am I audible?
Yes, you are audible.
Yeah, I am C22. Can I present?
Yes.
From session.
Yes, please start.
Sir, is my screen visible?
No, right now, not visible.
It is visible now?
No.
Sure. Ma'am, reshare your screen.
Yes, please share and please start.
She has given his thorough knowledge about the.
Sir, is it visible now?
Yes, visible now. Start.
Yeah, thank you, sir. So myself, Dr. Madhurima Juarda, I am the Assistant Professor of Department of Biotechnology from SRM Institute of Technology, Ramapuram, Chennai. Today I am going to present my work on a probabilistic approach on arsenic exposure levels through cooked rice, along with its benefits to appraisal from rural and urban populations of West Bengal. As we all know that arsenic contamination is of great concern in today's world, so we should be aware of everything that is going on in and around us. Apart from that, accumulation of arsenic in rice is a significant global concern with its severity varying based on rice cultivars and the degree of arsenic contamination in agricultural soil. The uptake of arsenic by rice plants is greatly influenced by the use of arsenic contaminated groundwater during cultivation period. Bengal Delta primarily depends on arsenic contaminated groundwater for agricultural purposes. Consequently, when food or crops are grown In the contaminated soil and water, this toxic metalloid finds its way to enter the food chain. As a result, the arsenic contamination in rice grain rice consumption poses a significant potential risk to human health. We can see that arsenic not only in cooked rice, but in rice grain, but also in cooked rice is not safe. So this work has been published in Environmental Science and Pollution Research, and where we have evaluated the arsenic content in cooked rice from cooking in contaminated water, along with distributions, as well as categorizing the risk factor and micronutrients, and providing that we have shown the benefit risk assessment. This study mainly focuses on the contribution of arsenic through cooked rice from rural and urban populations. The margin of exposure through cooked rice is less than 1, with significance or the existence of health risks for all the studied age groups. So we have calculated or we have collected the samples from 3 places: exposed, apparently control area, and control area. So where we have seen that the cooking process takes place under the arsenic contaminated rice and arsenic safe water, where we can see that the exposed area is highly exposed, but comparatively then comes the apparently control area and the control area. This percentage remains as usual distributed in the various way because the rice grain is being transported from the contaminated area to the uncontaminated area. So as a result, the arsenic is being transported to the human also living in the population, which is apparently control and control. So apparently control area show that the presence of inorganic arsenic is mostly dominated by raw rice along with the existence of a smaller amount of DMA in the cooked rice. So these are the categories of the inorganic arsenic and the organic arsenic present in rice grain. So two highlighting points from this results show the geographical variation in the combination of arsenic contaminated and uncontaminated rice before market. Here we can see that if we through a health risk exposure thermometer, that the apparent control and the control comes under the class 3, whereas for exposed it is class 4, which is highly exposed. So the margin of exposure through cooked rice is less than 1, which signifies the existence of health risk for all the studied population. From the next health risk appraisal, we can see that the life risk of cancer and the non-cancer risk. Sorry, the lifetime cancer risk analysis for the studied population through cooked rice is higher than the recommended value, whereas HQ, that is the value is greater than 1, has been observed for all the studied age groups from the exposed area and the adult male group from the control area. Adults and children from the rural area showed that the ingestion rate, that is the IR, and concentration and the respective influencing factors towards the rice arsenic, whereas the ingestion rate is solely responsible for the age groups from urban area. So these three differentiation, we get it from the Monte Carlo stimulation approach, that is the probabilistic approach, where this variation we have seen for the 3 different populations. Apart from this, a benefit risk assessment has been performed which supports that the selenium rich values in the cooked rice are effective in avoiding the toxic effect and the potential risk from the associated metal present in the cooked rice. So overall, we could suggest that the attempt should be undertaken to decrease arsenic accumulation in uncooked rice through the implementation of cultivation practices during the post-monsoonal season and improvement of post-harvesting practices. To avoid the arsenic-induced future health risk, a very important proposal is to reduce the ingestion rate of cooked rice for control populace, irrespective of the geographical area. The prevalence of health risk through rice poisoning should be exaggerated in near future. So apart from this, there is a mitigation option that arsenic safe water is only the process by which we can reduce the arsenic contamination scenario in all the exposed area and minimize the use of arsenic contaminated water for cultivation practices. So one such area where we find that a treatment plant, the surface water treatment plant, is only the way out which helps in reducing the arsenic exposure in the contaminated area. So overall, we can say that the arsenic exposure comes not only through water and cooking, from all dietary foodstuffs, mainly rice grain, leads to many cancerous, non-cancerous manifestations. So overall, safe drinking water and proper awareness and communication is required to reduce the arsenic toxicity pollution in the environment. So these are my publications, first authorships, my co-authorship publications, my research work, and I would like to thank everybody for please have a patient listening. Thank you.
Okay. Okay, good presentation, Madhurima.
Thank you, sir.
So can you tell me one, I have query actually. How
Yes,
can we
sir.
remove arsenic from the treatment plant? Can you explain?
Yeah, treatment plant is that we can have the arsenic removal treatment plants also. So, but apart from this is a surface water treatment plant which is done naturally. The water is collected and through sand filtration and different membrane filtration, the water is being filtered in a low level and it's been transported to the arsenic population free of cost so that they get less amount of arsenic exposure through the contaminated groundwater in that particular location. So this is totally a natural surface water treatment plant which I have studied in during my work. And arsenic removal plants is also there which needs to be monitored in the alternate 6 months so that the arsenic content can be checked.
Okay. So so how can we utilize that water for the irrigation? Is it accumulate in the plants, bodies or
We
fruiting?
cannot use that particular water for irrigation. We used to use the post-monsoonal water. So cultivation during post-monsoon season will help in the reduced accumulation of arsenic in the plant systems. So that when we use for pre-monsoon, we are using maximum of shallow groundwater, which will lead to a maximum accumulation of arsenic. To reduce that only, we would suggest that during a study we have performed, that post-monsoonal season is the best for cultivations. But when we use the surface water, then it needs to be produced in huge amount when we used to use it for cultivation purposes.
Oh, okay. Thank you, Dr. Madhurima.
Thank
So
you,
I
sir.
call to the next presenter. So anyone left here? Please let me know.
Sir, may I present?
Yes, sure. Meera Krishna, please. Please start.
Okay, sir.
So please share your PPT. Meera Krishna,
Sir,
please.
sir, I'm sharing.
Okay, so anyone left, please raise the hand who not given the presentation.
Hello, sir.
Yes.
Sir, I'll present later. I can't share the screen. Okay.
Why is it any issue? Go
Yeah.
with your arrow, share screen and share. So what kind of the issue you're facing? Please let me know.
Okay, okay.
So please try. So anyone else? Anyone else who not given presentation? Subhadeep, am I audible?
Yes, sir. Yes, sir.
So
I'm audible.
can you present? Please try to upload.
Yes, sir. Yes, sir.
शुभदीप प्लीज आप शुरू करिए तब तक वो प्रयास करती हैं करेंगी ठीक है
Yes, sir. Yes, sir.
प्लीज स्टार्ट
Sir, 1 minute, sir. Yes. I have an issue. Uh, yes, sir. But I'm audible, sir. Hello, I am audible, sir?
Yes, yes, you are audible. Please share. Please share. Please share your presentation or anything.
Yes, sir.
Your screen is there. Yes, please.
Yes, yes. Sorry for late, sir. Today I am pleased to present my research on enhancing the fertilizer quality of waterizing through the vermicomposting by the additions of biochar, conducted by addition to collaborations with Dr. Balachandran and Mr. Apurva Kole. Uh, Conventional fertilizers through the effective pose significant risks such as the soil depletion and water pollution. This has to lead a growing interest in the sustainable alternatives, vermicompost, and eco-friendly process, and transform the oriented waste and organic waste into the rich humus uh, under the anaerobic conditions, benefiting the plant growth and the soil health. Uh, this is the objective to evaluate the effect of different amount of biochar on the nutrient content in waterizing vermicompost, especially focused on the nitrogen, phosphate, and organic carbon by systematic analysis. Here we prepare the experiment for experimental design, Bharmi 1, 2, 3, 4, and here the compositions 1000 gram cow dung and 300 gram waterizing, and add the biochar 50, 100, 200, 300, and Bharmi C is the control. Now we uh, from here water has been sample was collected from the Gwalpara village, and Audang was collected from this here, and earthworm obtained from the Rothindo Tissue Vidyan Kendra Vishwavarti, and the earthworm is identified ZSI, and the earthworm is Isismia spirata. And here we use some method for pH. The method is electrode method, and EC for ion selective method, organic carbon Walke Black method, nitrogen Golden method, and phosphate Olsen method, and colony forming mix for port plate method. Here in the result part, we see pH in 60 days, the lowest amount of pH in Bharmi 2, and the highest amount of pH in V1, and 75 days, the lowest amount of pH in V2, and the highest amount of pH in V4, and in 90 days, the lowest amount of pH is VC, and Bharmi C, and the highest amount of pH in Bharmi 4. And another graph in electroconductivity, in 60 days, the lowest amount of pH in Bharmi C, and highest amount of pH in Bharmi 2, and 75 days, the lowest amount of electric conductivity in Bharmi 4, and highest amount of electric conductivity in Bharmi 2, and in 90 days.
Please conclude. Please conclude. Subdeep, please conclude.
Yes, sir. And total nitrogen in here, the total nitrogen in this graph, Bharmi 2 is the high, and OC, organic carbon, here highest amount of organic carbon in 60 days, V4, and 75 V4 is the highest, and 90 days the highest amount of organic carbon in V3, and here the carbon ratio in 60 days highest amount of carbon ratio in Bharmi 3, and 75 days highest amount of carbon ratio in 75 days V4, and 90 days highest amount of vermicompost is V4. Here, before inoculum of arthroarm, the colony forming amount is this 102, 123, 112, and when the arthroarm is then the colony forming are increased. In here, we see the principal component analysis relevant the variable such as nitrogen, phosphate, organic carbon, and organic matter, were primarily influence in the vermicomposting outcome. From in my conclude, the study under the scope that biofuel can not only enhance the nutrient availability and relevant the vermicompost, but also the optimize the compost quality for the agricultural use. I had a paper presentations we prepare and acknowledgement. Thank you.
Nandan and Meera, please share after that.
And thank you, sir.
Sir, I am Meera. May I present?
Yes, you can start.
Okay.
Okay, thank you, Jaideep. So, which species of the earthworm you use?
Sir, Isocinia fidata. Isocinia fidata.
Okay, okay, thank you. Okay, next, Meera.
So, good evening. I'm Meera and my colleague is Nandana. We are fourth year BTech students of TKM College of Engineering, Kerala. And I'm excited to present our research, which focuses on performance investigation on solar hybrid adsorption system for potable water generation. So, these are the contents. So, water scarcity is a critical issue worldwide, especially in areas with limited access to drinkable water. The solar hybrid adsorption system uses renewable energy to provide clean potable water from various sources, including seawater and wastes, making it ideal for water scarce areas. So, this system is sustainable and offers practical solution for regions facing water scarcity. Going to the motive for which we have selected this research, adsorption desalination system working on solar thermal energy suitable for purifying seawater has already been introduced. So our goal is to make it suitable for converting brackish water into potable water along with improving yield and efficiency. Going to the experimental setup. So this is a system layout and it is having two parts. Single bed adsorption desalination system and a humidifier-dehumidifier system. So the first one comprises of evaporator, adsorbent bed, and condenser. By absorbing heat from hot side of the thermoelectric cooler, the evaporator vaporizes and saline water, which then reaches on the adsorbent bed. The silica gel adsorbs the vapor, and the heat for adsorption is supplied by hot water using solar energy. When the vapor gets saturated, desorption occurs and vapor goes to the air-cooled condenser and we get the desalinated water. Humidifier-dehumidifier is used to improve the yield and efficiency. And next is the data that we have got while doing the experiment, and we have got the inferences that hot side temperature of the thermoelectric cooler affect adsorption and desorption process. The optimum value of hot side temperature that we got is 45 degrees Celsius, and this system has 48% more increase in water vapor adsorption than conventional adsorption desalination system. And going to the results, adsorption selection. That is, we have collected data of various adsorbents and tabulated them using MCDM and we ranked them. We got rank 1 as banana peel, but it has got many uses. That is like it is edible. So we have taken orange peel for our study. Now, how we have prepared the adsorbent material. To prepare the orange peel adsorbent, wash, dry, and use the peels, then treat them with ethanol and sodium hydroxide solution. Rinse, dry, and grind and sieve into powder. Finally, store the powder in an airtight container. So this is the way by which we have prepared the adsorbent. Then going to the composite preparation. To prepare the composite, the orange peel powder is mixed with silica gel using necessary binder. These are formed into pellets and dried. Finally, the conclusion. So solar hybrid system offers a sustainable, eco-friendly solution for producing clean water by utilizing solar energy and advanced adsorption methods to improve desalination efficiency. These are the references. Thank you.
Okay, so this is very good presentation. So can you please, I have one query regarding desalination. So can you explain the mechanism of the desalination? Whatever plant you share here.
So please.
Desalination, क्या ये adsorption से possible है क्या desalination?
Sir, I'm Vandana. I'm in this group.
Okay, no
Can I
issue.
explain?
Although it is good presentation, your plan of the presentation is very nice. So
Sir, can I explain what was
yes, please explain.
sir, do you want to explain about the system? Hello.
Yeah. Okay. So what I'm asking, adsorption can be remove the salinity from the water by the adsorption method?
No, adsorbent is used to remove the salinity from the water that we have collected. It collects the vapor and then it just go to the condenser and we get drinkable water from there.
Okay, it means heating and condensation is play main role for the desalination.
No, the adsorbent we use plays a major role.
Sorry,
The adsorbent that we use plays the major role.
which sorbent?
In the present system, we are using silica gel, but we are planning to use brackish water. So we are trying different adsorbents so that we can convert all different water into drinkable form.
Okay. Okay, thank you. So anyone who not given the presentation, please let me know who not given the presentation. Nandan, have you given? So I think everyone completed the presentation. Neha Sharma, Niharika, Mohini. So Niharika have done already. Mohini done. Megha, is Megha here? Kuldeep Soni here? Okay. Shakir Hussain. Swati Yadav here. So I think Sweta Saxena, last one candidate who not given. Sweta Saxena. Okay. Thank you everyone for your good presentation. So we will evaluate you and after that we'll decide what going to happen. So thank you very much for your presentation. So I am going to end this meeting. Thank you everyone.